In this video, we will discuss about how this microbial can influence the T cell differentiation and its function. So, before we directly go into this topic, let us quickly revise how this T cell functions. T cells get the antigen information from the antigen presenting cells, and thereby the activation of the T cells occurs depending on the antigen informations and can perform its respective functions. So it is essential that the antigen presenting cells is also been activated, and they are usually activated. This precursor form of these antigen presenting cells is the dendritic cells, which is the immature antigen presenting cells, which gets activated by this antigen informations is which gets activated through several ways, and some of the ways are shown here, and their inhibitory effects, that is to suppress antigen presenting cells, are also shown here. So how this, let us now go through our topic, that is how this microbial can influence the activations of these antigen presenting cells, and also the microbial can suppress the activated antigen presenting cells and the T cells. So first we will start with this inhibitory effect, and this inhibitory effect, one of the examples is this dietary fiber, which is then converted into small chain fatty acid. It can suppress the antigen presenting cells, as dietary fiber has known to be the or this small chain fatty acid has been shown that it can inhibit the activations of these antigen presenting cells. And also this primary bilic acid, which is converted into secondary bilic acids, then it can interact with this TGR5 FXR, and thereby it will also affect these antigen presenting cells and T cells interactions. And the other two compounds that are present in the microbials that are mentioned here is tryptophan and polyphenolic compound, and these two in together form this AHR that can suppress these T cells activations. On the other hand, there are also some of the microbials components that helps in the stimulatory effect of these antigen presenting cells and the T cells interactions. One of them is L-carnitine, choline, betainin. All these three helps in the formation of TMAO with the intermediate of the TMA. And at the same time, this flavonoids is also converted to DAT, which then helps in this APC and T cells interactions, thereby helps in the activations of the T cells. So this microbial has both the functions, that is to suppress, has the inhibitory effects against this T cells activations, and also some of the stimulatory effect that helps in the T cells activations, or that promotes the T cell activations. And how this small chain fatty acid, now we will understand how this small chain fatty acid helps in the suppressions of this antigen presenting cells. One of the ways it does is it sorry about that one of the ways that it does is it secretes this IL-10, and that well helps in the differentiation of the T cells into the regulatory T cells that will suppress the other antigen presenting cells. And this indole 3 lactic acid, and it will help in the secretion of IL-2, and that helps in the formations of this heterotoxic CD8 plus T cells. In other terms, it helps in the IL-2, IL-12 helps in the activations of the or helps in the differentiation of the T cells towards CD8 plus heterotoxic T cells. So basically what it does is it triggers the APC to produce this kind of cytokines. And in terms of this ATP, that is bacterially derived ATP, it helps in the formations of this IL-6, IL-23, and that will lead to the formation of TH17. And in other term, this stimulation is based on the microbials that can affect APC, and that leads to different kind of cytokine stimulations. And in terms of the plasma cells and epithelial cells, the small chain fatty acids also regulates or modulates these plasma cells to form interleukin 10 and modulates these epithelial cells to form TGF beta, IL-6, CXCL1, and this interleukin 10 then helps in the formations of these T regulatory cells, and the TGF beta, IL-6 and CXCL1 helps in the formation of TH17 and TH1 cells, and these are also the cytokines that are formed by the and these are the cytokines that are formed by non-APC cells. That is, these cytokines are formed by non-APC cells through this SCFA, small chain fatty acid interactions with plasma cells or epithelial cells. Sorry about that. And they can play their respective functions, such as regulatory T cells helps in the suppression of the activation of the T cells, and the TH17 and TH1 also has the pro-inflammatory and anti-inflammatory response, and thereby helps in the activations and suppressions of the T cells. So that's all for this video. I hope you understood in brief, this microbial can influence the T cell differentiation and its functions, and it's not just one way, but it will have both the effects, or the different kind of microbials has different kind of effects on the on this immune system, that is antigen presenting cells and the T cells activations and differentiations. That's all for this video. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.